very much to everyone for attending this webinar series. As you probably know, these series are held usually every other Thursday at 3:30. And it's been going on for a year and a half almost. And since last January, the seminars is open access and it's open to everyone who is interested in attending, whether they are part of CIBERNED or CIBER or any other institution. If you're interested in this seminar series, you can check in CIBERNED webpage the program for the following weeks. We have the program ready in the web up to next July, and we will post the following months in a few weeks' time. So, although probably most of you are familiar with the dynamics of this seminar series, let me just remind you a couple of things. First of all, today's seminar will be recorded. And as usual, when we record the webinars, we will post the webinar in the CIBERNED YouTube channel. Should be ready there for, I don't know, in the next few days. The seminar will last about 30 to 40 minutes, and then we will have some time for discussion. If you're interested in making a question, please send me a message through the chat option, just stating your name and affiliation, and I will give you the floor to make the question yourself in a first come, first serve basis, so we can have a more agile discussion this way. So there is no need for you to write down the question, since you're going to be able to do the questions directly yourself. I would acknowledge, we would like to ask everyone to keep their micros off, and the cameras are optional, but if you put it in off, probably there will be a better streaming of the webinar. Okay, so having said all that, I gotta introduce you today to our speaker for today's webinar. It's a pleasure to introduce you Marta Riba. Marta is a graduate in pharmacy for the University of Barcelona, where she has obtained her PhD just a couple of months ago, as I just learned. Her PhD has been performed in the group of blood-brain barrier, which is led by Drs. Carme Pellegrí and Jordi Vilaplana, the physiology section of the Faculty of Pharmacy at the University of Barcelona. She has had predoctoral grants from the Generalitat de Catalunya and also from the FPU, Ministerio de Universidades. And she's currently a postdoctoral researcher in the same group, who is linked to the Antonio Camins CIBERNED group. She has spent some pre-doctoral stays at the Dr. Matthew Gentry at the University of Kentucky, and she also been involved in some teaching in pharmacy and nutrition and food science and technology at the University of Barcelona. And without much delay, I would like just to thank Marta for her willingness to share with us her work, her thesis work. And her talk today is titled Wasteosomes, corpora amylacea, a physiological mechanism for the elimination of brain waste substances. So Marta, thank you very much. Welcome to the CIBERNED Webinar Series, and please go ahead with your talk. You can share your screen now and go ahead. Thank you.
Okay. Okay. Good afternoon, everyone. Thank you for introducing me, Dr. Medina. First of all, I'd like to thank CIBERNED for organizing these webinars and for allowing me to present our newest results today by means of this webinar entitled Wasteosomes, or also called corpora amylacea, a physiological mechanism for the elimination of brain substances. I will start talking about the main features of corpora amylacea in the human brain, just to contextualize. These structures were first described by Purkinje in 1837. They are spherical and measure 5 to 15 microns in diameter. They are based on carbohydrates or glycans, and that's why they are positive to periodic acid shift staining, as shown in this image. They are intracellular and are located in astrocytic processes, and they accumulate in the human brain during aging and accumulate also in specific areas of the brain in neurodegenerative conditions such as Alzheimer's disease or Parkinson's disease or other diseases such as obstructive sleep apnea. Regarding the composition of corpora amylacea, as I just mentioned, they are essentially based on glycans, mainly glucose. This carbohydrate or glycan structure is synthesized by glycogen synthase, an enzyme found in the limits of the corpora amylacea, as shown in this image in green. And we know that they have waste products from astrocytes, from oligodendrocytes, or neurons, for example, tau1 or tau5, and even fungal, bacterial, and viral proteins have been described in them. As you can see in this image, these bodies also contain ubiquitin in a central core and p62 in the peripheral zone. Both proteins are involved in processes of elimination of waste substances. And all these components and the way they are organized within the structure make us believe that corpora amylacea are carbohydrate-based structures that accumulate waste products. Moreover, corpora amylacea contain neoepitopes, but what are exactly these neoepitopes? They are epitopes that appear de novo in our bodies, such as in cell debris or apoptotic bodies, but we don't know exactly the nature of these neoepitopes. And curiously, these neoepitopes found in corpora amylacea are recognized by natural IgMs, and the interaction between the IgMs and the neoepitopes may give rise to an eat-me signal that leads to phagocytosis. That's why we think that somehow corpora amylacea may be phagocytosed by macrophages. However, these IgMs cannot reach the central nervous system in physiological conditions. Due to the blood-brain barrier, these IgMs cannot cross it. So, besides corpora amylacea are intracellular, they are found in the astrocytic processes. Thus, the interaction between corpora amylacea and IgMs would take place in case of a blood-brain barrier disruption and a release of the corpora amylacea outside the astrocytes, or in case of release of the corpora amylacea outside the central nervous system. So it seems that physiologically this second option would be more plausible and which is supported by the location of corpora amylacea. These corpora amylacea are located in the brain interfaces, mainly in the periventricular, subpial, or perivascular regions. All these regions are close to or connected to the cerebrospinal fluid, and that's why we propose that corpora amylacea could be released from the brain tissue to the cerebrospinal fluid. We know that cerebrospinal fluid drains not only through the arachnoid granulations, as classically believed, but also through the meningeal lymphatic system. So if corpora amylacea were released to the cerebrospinal fluid, we believe that they could not cross the arachnoid granulations and access the blood because of their size. They are too large, but they could access the meningeal lymphatic system. This figure shows the pathway of a corpora amylacea that would be extruded from the brain tissue to the cerebrospinal fluid of the subarachnoidal space, where there are some lymphatic capillaries. So the corpora amylacea from this space could access these capillaries and reach the lymphatic vessels and finally the lymph nodes, where they may be phagocytosed by macrophages. So up to this point, we consider that corpora amylacea accumulate waste products from the brain. They contain neoepitopes that may give rise to the phagocytosis. According to their location, they may be released to the cerebrospinal fluid, and from this cerebrospinal fluid, they could access the meningeal lymphatic system and reach the lymph nodes, where actually they may interact with macrophages and be phagocytosed. In this regard, our objectives were to determine the presence of corpora amylacea in the cerebrospinal fluid and in the cervical lymph nodes. We plan to study the interactions between corpora amylacea and macrophages, and we tried to determine the nature of the neoepitopes of corpora amylacea, which is unknown so far. So regarding the first objectives, to figure out if there were corpora amylacea in the cerebrospinal fluid, we got human ventricular postmortem cerebrospinal fluid, and we stained the samples with PAS staining, with an antibody against glycogen synthase with IgMs, and we processed the samples by transmission electron microscopy and scanning electron microscopy. All these techniques allowed us to observe these spherical structures that showed the same histological and immunohistochemical features and the same ultrastructures as the corpora amylacea from the hippocampus. So we confirmed that human cerebrospinal fluid contains corpora amylacea. Moving on to the second objective, we tried to find out if there were corpora amylacea in the cervical lymph nodes. In the same way as cerebrospinal fluid, we got sections of human cervical lymph nodes and we stained them with PAS staining and with IgMs. As you can see in these pictures, we observed that some corpora amylacea in these sections were present. Therefore, we confirm that there are corpora amylacea in the cervical lymph nodes. Particularly, these corpora amylacea in the cervical lymph nodes were located in areas where the lymph flows, particularly in trabecular sinuses or in trabecular or subcapsular space. So even we saw corpora amylacea showing a specific context with lymph node cells that could be macrophages or other cells. In addition, we quantified the number of corpora amylacea in the lymph nodes according the location of these nodes in the upper zone, the intermediate zone, and the lower zone of the neck. And we saw that there were more corpora amylacea in the upper regions compared to the lower regions, which revealed that the lymph nodes would act as a filter, retaining and eliminating corpora amylacea. Regarding the third objective, we studied the interactions between corpora amylacea and macrophages. So we purified corpora amylacea from the cerebrospinal fluid, we opsonized them with IgMs, and we incubated them in a THP1 macrophage cell culture to study this interaction. Then we processed the samples using PAS staining, immunofluorescence, and scanning electron microscopy. In all these experiments, we saw macrophages making contact or surrounding corpora amylacea. Or even in this case, we saw a corpora amylacea inside a macrophage. Thus, it's clear that THP1 macrophages can phagocytose corpora amylacea opsonized with IgMs. But surprisingly, control experiments without IgMs showed the same results. We observed corpora amylacea without IgMs surrounded by macrophages. So other mechanisms apart from IgMs could lead to the phagocytosis of corpora amylacea. According to these results, we decided to study the whole phagocytosis process, and we stained the macrophages with a cell tracer, and we stained the corpora amylacea with concanavalin A, and we perform a time lapse. I will show some videos now. In this first video, we can see a corpora amylacea in red, orange, and the cells in green. We can see how this cell captures the corpora amylacea and internalizes it and begins to degrade it until the corpora amylacea is mostly digested. We can see in this confocal microscopy image, we can observe the fluorescent particles coming from the degradation of the corpora amylacea located in the surface of the cell. In this other video, we can observe a larger corpora amylacea in red that is attacked by several cells at the same time, and we can see how these cells start breaking up the corpora amylacea in small particles, and we can notice that they capture these particles and accumulate them inside. However, these macrophages cannot fully digest the corpora amylacea, probably because of the large size of the structure. And in this third video, we can see 1, 2, and 3 corpora amylacea. I ask you to pay attention to this one. We can notice that this cell ends up breaking up a large piece of the corpora amylacea and take it. So certainly, corpora amylacea labeled with concanavalin interact with macrophages. The smallest ones are phagocytosed completely and digested by one cell, and the largest ones are attacked by several cells at the same time, but not completely digest. However, we should consider that concanavalin is a vegetable protein that could be recognized as an immunogenic particle and trigger the phagocytosis by itself. So what we did was directly label the proteins of the corpora amylacea themselves with the NHS ester proof that conjugates directly the proteins, and we performed a time lapse too. In this first video, we can see a corpora amylacea in red, orange, and a cell that binds it and starts moving it. Now we can see how the cell fragments the corpora amylacea here, and we can see these small particles, and now we know that these particles come from the corpora amylacea itself. This second video shows the corpora amylacea being attacked by different cells which degraded and internalized some particles as well. Thus, thanks to these videos, we observed that corpora amylacea interact with macrophages, undergo degradation processes, and by themselves without IgMs or concanavalin or other particles that may stimulate the phagocytosis. At this point, we move to the fourth and last objective with the neoepitopes. We tried to figure out the nature of the neoepitopes of corpora amylacea. Just an aside before continuing, do you remember that I mentioned that corpora amylacea have neoepitopes that are recognized by natural IgMs, but the nature or the composition of these neoepitopes is unknown? Well, we tried to determine the nature of these neoepitopes. And well, we know that carbohydrates and proteins are the main components of the corpora amylacea. So in a first place, we wondered if the neoepitopes of the corpora amylacea were of a protein nature. To answer this question, we applied the following strategy. We purified corpora amylacea from the cerebrospinal fluid. We added pepsin, which is an enzyme that breaks down proteins. And then we incubated IgMs. Remember that these IgMs recognize neoepitopes. And if the neoepitopes were proteins, we would expect that the pepsin would digest and eliminate the neoepitopes, and therefore we would not see staining. Otherwise, if the neoepitopes were not proteins, they would remain intact against the pepsin digestion. Thus, the IgMs could bind to the neoepitopes, and we would see the staining. To validate this method, we applied the same strategy. We purified corpora amylacea from the cerebrospinal fluid, we digest them with pepsin, and we incubated an antibody against the protein P6C2, and pepsin should digest the protein P6C2, so that we expected not to see the staining. On the other hand, to make sure that pepsin was the only was affecting only the proteins and not other structures, we stained the corpora amylacea digested with pepsin with concanavalin A. That binds to glycans, not proteins. So we would expect that the staining would not be affected by the pepsin treatment. Well, these figures show the results we got. In the upper figure, we see in the first row the control without pepsin, the same corpora amylacea stained with concanavalin and with P6C2 and the merged images. In this lower row, we can observe a corpora amylacea digested with pepsin stained with concanavalin, but not with P62, which demonstrates that the enzyme works and digests the proteins and glycans are not affected. In this lower figure, we can observe in the upper row the control without pepsin with the same corpora amylacea stained with IgMs and P62. And in the lower row, we can see a corpora amylacea digested with pepsin that is not stained with P62 and it's stained with IgMs, which indicates that neoepitopes are not proteins. So if they are not proteins, as I've just mentioned, the main components of corpora amylacea are glycans and proteins. So we wondered if the neoepitopes of the corpora amylacea were glycans. Accordingly, we applied the following strategy. We know that if we apply IgMs in a section of the hippocampus, these IgMs recognize the neoepitopes of the corpora amylacea. And if we add a secondary antibody against the IgMs, we observe a fluorescent labeled corpora amylacea. So if we consider the neoepitopes as a glycans, it means that IgMs recognize glycans. So the glycan recognition domain should be affected if we add monosaccharides to the IgM solution. Well, we prepared different concentrations of these monosaccharides and we added them to the IgM solution, expecting that these monosaccharides would block the binding domain of the IgMs and they can no longer bind to the neoepitopes of the corpora amylacea. Thus, we would expect that the corpora amylacea would not be stained. To validate this method, we applied the same strategy using concanavalin and P62. On the one hand, concanavalin, which recognizes glycans, we would expect that the addition of monosaccharides would interfere with the staining with concanavalin. On the other hand, using the antibody against P62, which we would expect that it would not be affected with the addition of monosaccharides, and these results confirm that these assumptions were correct. So we confirmed that that was an accurate method for assessing interactions based on carbohydrate recognition. And what happened with IgMs? In this figure, we can see each column with monosaccharide and each row with the different concentrations we tasted. Let's focus on glucose. We can notice that at 0 millimolar concentration of glucose, we can see the corpora amylacea stained with IgMs. And apparently there's a decrease in the staining with increasing concentrations of glucose. We can observe the same effect with the other monosaccharides, such as galactose, fructose, and mannose. Moreover, we quantified the intensity of fluorescence and we observed that there was a progressive decrease in the staining with increasing concentrations of all sugars stained, which indicated that the sugars tested interfere with the interaction between IgMs and the neoepitopes. And that's why we think that neoepitopes of corpora amylacea have a glycan nature. So to sum up, we demonstrated that there are corpora amylacea in the cerebrospinal fluid, in the lymph nodes, that corpora amylacea can be phagocytosed by macrophages, and the neoepitopes of corpora amylacea have a glycan nature. So we conclude that corpora amylacea, which are structures that accumulate waste products from the brain, are expelled from the brain tissue to the cerebrospinal fluid and reach the lymph nodes through the meningeal lymphatic system. We also observe that macrophages can phagocytose corpora amylacea in vitro. That's why we point out that corpora amylacea can be phagocytosed in the lymph nodes by different mechanisms apart from the interaction between a glycan neoepitope and IgMs. So taken together, we conclude that corpora amylacea can act as containers that remove waste substances from the brain. I'd like to mention that corpora amylacea can be found in other organs or tissues apart from the brain, such as the lung, breast, uterus, prostate. All these corpora amylacea from different organs share some histological and ultrastructural features, such as PAS staining or other stainings, and they accumulate waste products too. Even some authors describe that they interact with macrophages as well. So we extrapolated this function that we given to the corpora amylacea of the human brain as waste containers to the corpora amylacea of other organs and tissues. and we propose to rename these structures as Wasteosomes. Accordingly, we proposed a new hypothesis that support a new mechanism for the elimination of waste substances from the human body, which we have called the Wasteosome system. We know that different factors such as age, oxidative stress, external particles, or infectious agents produce waste substances. These substances can be removed by autophagy processes and lysosomes, or the ubiquitin proteasome system, or the Wasteosome system. This system involves the ubiquitin MP62 as an adapter molecules which collect waste products. And thanks to several enzymes of the glycogen metabolism that synthesize the glycan structures, such as glycogen synthase, the waste products would accumulate inside the glycan structure. In addition, because of the waste substances or the glycan structure, these corpora amylacea or wasteosomes generate new epitopes that may act as immune signals. And then corpora amylacea would be extruded or expelled to the external media or to the interstitial spaces where macrophages would phagocytose and eliminate the corpora amylacea or wasteosomes. So most of these findings are compiled in these 3 articles, and they are very interesting, and I invite you to read them. And well, finally, I should acknowledge the Ministerio de Economía, the Ministerio de Universidades, el Institut de Neurociències de la Universitat de Barcelona, la Generalitat de Catalunya for all the funding, and special thanks to the electron microscopy and advanced optical microscopy services for their help, and the Neurological Tissue Bank for supplying brain samples. Thanks to TFEB Research Group, who taught us the cell culture techniques. And thanks to CIBERNED and Antónica Means for giving me the opportunity to present today. And I would acknowledge Carme Jordi and Jaume del Valle for their support, their advice, willingness, and guidance. And now, if you have some questions, I will be happy to answer them. Thank you very much.
All right, thank you very much, Marta, for a nice presentation. So now the paper is open for questions. So as I said earlier, if you're interested in making a questions, please do send me a message through the chat or raise your hands and state your name and affiliation, and I will give you the floor to make the question yourself. I see we already have one request here. Let me see if I get. Yeah, we have a first question from Ramon Trullas. Ramon, please go ahead.
Hello, Marta, thank you very much. This is very nice presentation, extremely interesting data. Thank you very much. I wonder whether there is a difference between astrocytes, or you know any difference between astrocytes and neurons in their ability to produce or release the corpora amylacea.
Well, we think that corpora amylacea are only generated inside the astrocytes. So we think that the astrocytes are the only cells with the ability to extrude this corpora amylacea. However, these corpora amylacea have proteins from astrocytes neurons, so we think that astrocytes collect some cell debris or neurons in degeneration and integrate some waste substances coming from these neurons in degeneration inside the structure of the corpora amylacea.
So the interpretation of the neuronal proteins that the corpora amylacea contain are just from waste outside the astrocytes or from products taken up by the astrocytes.
Exactly. Our hypothesis is that the astrocytes captures the neuronal proteins and integrate them in the corpora amylacea.
And just one last question. Is there any difference in or regional preference for the production of corpora amylacea in the brain?
Yes. we think that, well, we believe that corpora amylacea accumulate in the peripheral areas of the brain, close to pia mater, close to ventricles, and close to vessels. And we think that this location is involved in the extrusion of the corpora amylacea outside the brain. We think that there's like a moving of waste products to the periphery of the brain, and then there's an accumulation in these regions.
Okay, thank you. Thank you very much.
Thank you. Thank you.
All right, thank you, Ramon. Thank you, Marta. We have another question from, in this case, from Estrella Rausel from the Universidad Autónoma de Madrid. Estrella.
Hello, Marta. Thank you very much for your excellent presentation. It's been very, very clear and very nice presentation. I would like to ask you if you know if corpora amylacea are also present in other primates like monkeys or rodents?
I think so. I think corpora amylacea are described in primates, and in rodents, in mice, they are called pus granules or corpora amylacea-like granules. And we think that they have a different distribution. They are polyglucosan bodies as well, but they have like some different features in some ways.
Mm-hmm. Thank you. So they have been described, but they have like a different nature, correct? And they also come from astrocytes. They are also released from astrocytes in primates and rodents.
We think so, but I'm not sure about that.
And related to this, I'm sorry because I'm completely ignorant about this matter. But how do you know? How do you know these corpora amylacea are released by astrocytes?
Well, they are formed inside the astrocytes. We have some confocal images and electron microscopy images that locate these structures inside the astrocytic processes. So we are not sure. We haven't seen that, but is the more plausible explanation. And
so related with the previous question, the membranes of the corpora amylacea, could they still have channels or proteins that were existing previously in the astrocyte?
It could be. We don't know it, but it's possible. It's possible. Why not?
Mm-hmm. And the last question, I'm sorry, I don't want to make this very long, but why would the brain wanted to waste the glycans?
How? Sorry?
I mean, see, you mentioned that the contents of the wasteosomes are mainly glycans.
Uh-huh.
Why would the brain would like to get rid of those glycans?
We think that the glycans are the structure of the corpora amylacea, but they are not the waste. We think that the glycans constitute the container, which within well, the glycan structure accumulates the waste inside.
So it's mainly the membrane of the corpora amylacea is made up of glycans, correct?
Correct.
It's to which IgM is joining, correct? But it also has properties of any other membrane, like lipids and things like that, correct?
Could be.
You don't know, okay.
I don't know.
Lots of things to know. Thank you very much. And
no, thank you for your very interesting questions. Thank you.
All right. Thank you, Estrella. Thank you, Marta. We have another question, this one from Joanna Garcia. Please, Joanna, you can pose your question yourself.
A question regarding the interaction between corpora amylacea and microglia. Since microglia can phagocytose debris and so on, is able to phagocytose corpora amylacea in the parenchyma?
We haven't studied the interaction between corpora amylacea and microglia, but I would say that corpora amylacea are intracellular in the central nervous system, so I think that they would not interact, but we saw recently interactions between corpora amylacea and macrophages in the brain interfaces, but they were not microglia. They were just macrophages in the brain interfaces, in the meninges or in the choroid plexus, and they were interacting with corpora amylacea that were released from the brain tissue.
Okay, thank you.
Thank you.
All right, thank you, Joanna and Marta. So we have some more time for questions if there are anyone interested. As I said, if that's the case, please just send a message or just raise your hand. Okay. I just got another request here. This one is from Ju Yang Li from MPI for Biological Cybernetics. So please go ahead and make your questions.
I hope you can hear me. Thank you very much for your presentation. I had a question about this hypothesis that corpora amylacea generated in the brain travels through meningeal lymphatic system and then ends up in the lymph node in the neck region. Is it possible that corpora amylacea found in this lymph node are just innately produced in lymph node, but not necessarily, like you've mentioned that corpora amylacea are found in breast and other organs as well. So could it be that it is just generated inside the lymph node and then not necessarily coming from the brain?
This is a very interesting question. We believe that the corpora amylacea in the lymph nodes come from the brain because they are recognized by natural IgMs. And this is a feature that only have the corpora amylacea from the brain, not from the other tissues. We think that, well, we should perform more assays to confirm this, but this is the main reason why we believe that corpora amylacea in the lymph nodes come from the brain and they are not generated in the lymph nodes. And moreover, they are located in subcapsular space or trabecular sinuses where the lymph flows. So we think that the regions where corpora amylacea are located in the lymph nodes are related to this flow of the lymph through the brain towards the lymph node.
I see. Thank you.
Oh, thank you. Very interesting question.
All right. Thank you both. So we have one more question here. This one is coming from Marcos Galan. Marcos, go ahead.
Hi, hello. Thank you very much, Marta, for your great presentation. Very, very interested. So congratulations on that. And I was wondering, since you said that these corpora amylacea accumulate, for example, through aging or in neurodegeneration, if they are formed inside the astrocytes, have you seen whether this accumulation of corpora amylacea in the astrocytes may lead to some kind of degeneration of the astrocytes or loss of function of these astrocytes? Do they die because of the accumulation of corpora amylacea or not?
Sincerely, we don't know. I don't know. I'm sorry,
Okay.
I cannot ask this question. I'm not sure. Maybe, well, this glycan structure protects the astrocyte from the action of these waste substances and maybe protect the astrocytes, but I'm not sure about that. It's only a hypothesis.
Okay, thank you. Thank you very much, Marta.
All right, thanks, Marcos and Marta. Are there any other questions? I don't see any other requests for questions. You wait for a few more seconds just in case.
If I may, I raise my question. May I ask a second question?
Yes, please, go ahead.
Marta, this is Estrella again. Just one more question. Is any astrocyte releasing corpora amylacea anytime, or do they need any kind of a stimulus to release corpora amylacea?
Actually, I'm not sure about that, but I would say that the accumulation of many corpora amylacea produces the release of them to the cerebrospinal fluid. But maybe the accumulation of waste, the accumulation of neurodegeneration in some areas produces more release, but I'm not so sure.
Okay, thank you. Yeah, I need.
Thank you.
All right, thank you again. All right, so this is the last call for questions. If there is someone else still interested in there, yes, we have again Yu Young Lee. Please go ahead.
Hi, I'll make the use of the last minutes. I read in literature that corpora amylacea can vary in their sizes. So it can be as big as 30 micrometer diameter, right? So do you think astrocytes, like these large corpora amylacea generates within astrocytes, like and then it gets exposed outside the cell, or is it possible that the corpora amylacea becomes bigger in the extracellular space?
I think these corpora amylacea become bigger inside the cell, and when the corpora amylacea is extruded outside the brain tissue in the cerebrospinal fluid, then they may interact with some cells or whatever. But in any case, we got some images from electron microscopy, and these corpora amylacea were surrounded by a membrane that was from the astrocyte. So it was pretty clear that these structure were inside the cell.
Okay.
In the brain tissue.
Yep. Thank you.
Thank you.
All right, thank you. So if there are no more questions, and I don't see any other coming either with the raise hand or through the chat, then I think it's time to close the session. Not before thanking Marta for a very nice presentation.
Thank you all.
Really fruitful discussion as well. Thank you very much for attending, and wish you all a very nice afternoon. I'll see you in hopefully a couple of weeks. Bye-bye.
Bye. Thank you. Thank you all.